Hey everyone, welcome to Advanced Exercise Physiology, Chapter 7, the Nervous System, the Structure and Control of Movement. The objective of this chapter will be 1. To discuss the general organization of the nervous system, 2. To describe the structure and function of a nerve, 3. To draw and label the pathways involved in a withdrawal reflex, 4. To define depolarization, action potential, and repolarization. 5. To discuss the role of a position receptors in the control of movement. 6. To describe the role of the vestibular apparatus in maintaining equilibrium. 7. To discuss the brain centers involved in voluntary control of movement. 8. To describe the structure and function of the autonomic nervous system. Now this is a simple outline that you can follow if you're taking notes. There are several major categories, so please take the time to write them down and organize yourself before the lecture begins. Starting with the general nervous system functions. It controls the internal environment with the endocrine system. It controls the voluntary control of movement. It also is involved in the programming of spinal cord reflexes and a simulation of experiences necessary for memory and learning. If we look at the organization of the nervous system, it is made of two components. The central nervous system, which is comprised of the brain and spinal cord, and the peripheral nervous system, which are neurons outside the central nervous system. There is a sensory division, where afferent fibers transmit impulses from the receptors of the central nervous system, and a motor division, where efferent fibers transmit impulses from the central nervous system to effector organs. Below you'll see a diagram of the anatomical divisions of the nervous system. In addition, you can see the relationship between the peripheral nervous system and the central nervous system. Both of these diagrams are present in your textbook. In summary, the nervous system is the body's means of perceiving and responding to events in the internal and external environments. Receptors capable of sensing things such as touch, pain, temperature, and chemical stimuli send information to the central nervous system concerning changes in our environment. The central nervous system responds by either voluntary movement or a change in the rate of release of some hormone from the endocrine system, depending on which response is appropriate. The nervous system is divided into two major divisions, the central nervous system and the peripheral nervous system. The central nervous system includes the brain and spinal cord, whereas the peripheral nervous system includes nerves outside the central nervous system. Moving on, we want to discuss the structure of a neuron. A neuron is composed of a cell body, which contains the nucleus, dendrites, which conduct impulses toward the cell body, an axon, which carries electrical impulses away from the cell body, and may be covered with Schwann cells, which forms discontinuous myelin sheath along the length of the axon, as well as a synapse, which is contact points between the axon of one neuron and the dendrite of another neuron. This is a diagram showing you the parts of a neuron. Now looking at multiple sclerosis and the nervous system function. Multiple sclerosis is a neurological disease that destroys the myelin sheaths of the axon. It has a genetic component, and it is due to an immune attack on myelin. It results in progressive loss of nervous system function, which include fatigue, muscle weakness, poor motor control, loss of balance, and mental depression. It is found that exercise can improve functional capacity, which leads to an improved quality of life. Next, you will see a diagram of synaptic transmission, where the axon of one neuron is in contact with the dendrite of another. Now, if we discuss the electrical activity in neurons, we see that neurons are an excitable tissue. They have an irritability, which is the ability to respond to a stimulus and convert it to a neural impulse. We also see that they have a conductivity, which is the transmission of the impulse along the axon. Part of the neuron involves a resting membrane potential. What we see is a negative charge inside the cell at rest, which is -5 to -100 millivolts, and -40 to -75 in neurons. This is determined by the permeability of the plasma membrane to ions, as well as the difference in ion concentrations across the membrane, which include sodium, potassium, chloride, and calcium. This is maintained by a sodium-potassium pump, where potassium tends to diffuse out of the cell. The sodium-potassium pump moves 2 potassium in and 3 sodium out of the cell. This is a diagram of the resting membrane potential in the cell and shows the difference across the membrane. Next, we see the concentration of each of the ions involved across the cell membrane. One involves intracellular fluid concentrations inside the cell. The other involves the extracellular fluid concentrations outside the cell. Next, we see an illustration of the ion channels that are involved in maintaining that membrane potential. Most importantly, the sodium-potassium pump plays a crucial role in the maintenance of the membrane potential. Next, we would like to discuss the action potential of a neuron. This occurs when a stimulus of sufficient strength depolarizes the cell. What happens then is there's an opening of the sodium channels, and sodium diffuses into the cell. What results is the inside of the cell becomes more positive. Following that, we see repolarization, which is a return to resting membrane potential. Potassium leaves the cell rapidly, and the sodium channels close. This is an all or none law. Once a nerve pulse is initiated, it will travel the length of the neuron. Here we see a diagram of the action potential, and then following repolarization to the original resting potential. Within each action potential, we have a depolarization and a repolarization of the nerve fiber seen in the diagram below. Please reference your book for both of these figures. Moving on and discussing neurotransmitters and the synaptic transmission, we see at the synapse a small gap between presynaptic neurons and postsynaptic neurons. Neurotransmitters are chemical messengers released from the presynaptic membrane, which then bind to a receptor on the postsynaptic membrane. This causes depolarization of the postsynaptic membrane. Here you will see the basic structure of a chemical synapse between the presynaptic and postsynaptic membranes, and also see how neurotransmitters cross the barrier. Again, talking about neurotransmitters and the synaptic transmission, we have an excitatory postsynaptic potential. This causes the depolarization. In addition, there's a temporal summation, the summing of several EPSPs from one presynaptic neuron. There's also a spatial summation, which is the summing from several different presynaptic neurons. In addition, we have an inhibitory postsynaptic potential caused by hyperpolarization. In summary, nerve cells are called neurons and are divided anatomically into 3 parts: the cell body, dendrites, and the axon. Axons are generally covered by Schwann cells, with gaps between these cells called nodes of Rondeer. Neurons are specialized cells that respond to physical or chemical changes in their environment. At rest, the neurons are negatively charged in the interior with respect to the electrical charge outside the cell. The difference in the electrical charge is called the resting membrane potential. The neuron fires when a stimulus changes the permeability of the membrane, allowing sodium to enter at a high rate, which depolarizes the cell. When the depolarization reaches threshold, an action potential or nerve impulse is initiated. Repolarization occurs immediately following depolarization due to an increase in membrane permeability to potassium and a decreased permeability to sodium. Neurons communicate with other neurons at junctions called synapses. The synaptic transmission occurs when a sufficient amount of a specific neurotransmitter are released from the presynaptic neuron. Upon release, the neurotransmitter binds to a receptor on the postsynaptic membrane. Neurotransmitters can be excitatory or inhibitory. An excitatory neurotransmitter increases neuronal permeability to sodium and results in an excitatory postsynaptic potentials. The inhibitory neurotransmitters cause the neurons to become more negative or hyperpolarized. This hyperpolarization of the membrane is called the inhibitory postsynaptic potential. Now looking at sensory information and reflexes. We have proprioceptors, which are receptors that provide the central nervous system with information about the body's position. These are located in joints and in muscles. We also can talk about kinesthesia, which is a conscious recognition of the position of body parts, and is also involved in limb movement rates. Now to discuss joint proprioceptors, we have free nerve endings, which are sensitive to both touch and pressure. They initially are strongly stimulated, but then adapt. We also have Golgi-type receptors, which are found in ligaments and around joints. These are similar to have free nerve endings. We also can talk about Purkinje corpuscles, which are found in tissues around joints. They detect the rate of joint rotation. Now, if we discuss muscle proprioceptors, they provide sensory feedback to the nervous system, which would involve tension development by a muscle and account for muscle length. There's also muscle spindles and Golgi tendon organs. The muscle spindle responds to a change in the muscle length. These consist of intrafusal fibers, which run parallel to normal muscle fibers, called extrafusal fibers. There's also gamma motor neurons, which stimulate intrafusal fibers to contract with extrafusal fibers by alpha motor neurons. In addition, there's a stretch reflex, which happens when stress on the muscle causes a reflex contraction. An example of this would be the knee-jerk reflex. Below you will see a diagram which more in detail shows the structure of the muscle spindles. Again, this is in your book to further review. Now to discuss the Golgi tendon organ, or GTO, we have an organ that monitors tension developed in a muscle. It also prevents the muscle damage during excessive force generation. What we see is stimulation resulting in a reflex relaxation of the muscle. We have inhibitory neurons that send IPSPs to each muscle fiber. We also see the ability to voluntarily oppose GTO inhibition may be related to gains in strength. And here is a more detailed diagram of the Golgi tendon organ, which is also seen in your textbook. Moving on to muscle chemoreceptors, we see they are sensitive to changes in the chemical environment surrounding the muscle, such as hydrogen ions, carbon dioxide, or potassium. They also provide the central nervous system with information about metabolic rate of muscular activity. They're very important in the regulation of cardiovascular and pulmonary responses. Next, we also see that the body has a withdrawal reflex. This is a reflex contraction of skeletal muscle. It occurs in response to a sensory input, but it is not dependent on higher brain centers. The pathways are neural reflex. The sensory nerve sends impulses to the spinal column. Interneurons activate motor neurons. The motor neurons control movement of the muscles. We also see in this reciprocal inhibition, where EPSPs to muscles to withdraw from stimulus, and IPSPs to antagonistic muscles. Finally, we see crossed-extensor reflex, where opposite limb supports the body during the withdrawal of the injured limb. Here's another diagram of the crossed-extensor reflex. In summary, proprioceptors are position receptors located in joint capsules, ligaments, and muscles. The three most abundant joint and ligament receptors are free nerve endings, Golgi-type receptors, and Pacian corpuscles. These receptors provide the body with a conscious means of recognizing the orientation of body parts as well as feedback about the rates of limb movement. Muscle chemoreceptors are sensitive to changes in the chemical environment surrounding the muscle and send information back to the central nervous system about the metabolic rate of muscular activity. We also saw that muscle spindles function as a length detector in muscles. In addition, Golgi tendon organs continuously monitor the tension developed during muscular contraction. In essence, Golgi tendon organs serve as a safety device that help prevent excessive force during muscle contractions. Also, reflexes provide the body with a rapid, unconscious means of reacting to some stimuli. Next, I'd like to discuss somatic motor function. This is controlled by somatic motor neurons of the peripheral nervous system. These neurons are responsible for carrying neural messages from the spinal cord to skeletal muscle. Within each motor unit, we have a motor neuron and all its muscle fibers that it innervates. Now, looking at the innervation ratio, we are going to discuss the number of muscle fibers per motor neuron. We see a low ratio in muscles that require fine motor control, approximately 23:1 in extraocular muscles. In contrast, we see a very high ratio in other muscles, approximately 1,000:1 or greater in the larger muscle groups. This is an illustration of the motor unit. Now, looking at motor unit recruitment and the size principle, what we see is during motor unit recruitment, there's a recruitment of more muscle fibers through motor unit activation. The size principle is the smallest motor units recruit first, produce larger EPSP, and result in action potential sooner. There are different types of motor units. We have type S, or slow, which sometimes are called type 1 fibers, and the smallest ones. We have type FR, which is fast, fatigue resistant, or type 2A fibers, sometimes considered intermediate. And we have type FF, fast and fatigable, or type 2X fibers, which are the largest. The recruitment pattern during incremental exercise is type S, type FR, and type FF. In summary, the somatic motor portion of the peripheral nervous system is responsible for carrying neural messages from the spinal cord to the skeletal muscle fibers. A motor neuron and all the muscle fibers that innervates are known as a motor unit. The number of muscle fibers innervated by a single motor neuron is called the innervation ratio. Finally, the size principle is defined as the progressive recruitment of motor units beginning with the smallest motor units and progressing to the larger and larger motor neurons. Now, moving on to the vestibular apparatus, this is located in the inner ear and is responsible for maintaining general equilibrium and balance. It also maintains head position. It is sensitive to changes in linear and angular acceleration. and is also stimulated by head movement. Finally, this vestibular apparatus also controls head and eye movement during exercise. Below you can see all of the roles of the vestibular apparatus in maintaining equilibrium and balance. In summary, the vestibular apparatus is responsible for maintaining general equilibrium and is located in the inner ear. Specifically, these receptors provide information about linear and angular acceleration. Now we want to talk about the brainstem. The brainstem is responsible for many metabolic functions. It is also involved in cardiorespiratory control and complex reflexes. The major structures include the medulla, the pons, the midbrain, and the reticular formation. The cerebellum includes the cerebral cortex, which is an organization of complex movement, storage of learned experiences, and reception of sensory information. The motor cortex is involved in motor control and voluntary movement. The cerebellum also coordinates and monitors complex movement, which incorporates feedback from proprioceptors. It also has connections to the motor cortex, the brainstem, and the spinal cord, and it initiates fast ballistic movements. Now, relating to sports-related concussions, we see that brain injury is a result of traumatic force. However, most of this typically resolves within a few days and may or may not involve the loss of consciousness. However, this all may result in permanent brain damage or death from delayed brain swelling, a second impact syndrome, where we have a second head injury before the first is healed, and this leads to cerebral swelling and death. We also can see this in same-season repeat concussions, and there are also late-life consequences of repeated concussions, such as Alzheimer's disease. Below you can see a list of different sports-related concussions. And also we see a diagram involving the cerebellum, the cerebrum, and the brainstem. Next, I want to discuss the role of the brain in exercise-induced fatigue. We have essential fatigue, where higher brain function centers and/or motor neurons happens. We have a depletion of the excitatory neurotransmitters in the motor cortex during this time, and a reduced motor output to the muscles as a result. This would involve potentially the central governor theory. This involves central control center regulating exercise performance, where we would see a reduced motor output to the exercising muscles. This protects against catastrophic disruptions of homeostasis, but more research is needed on this mechanism. What we see in current research is that it suggests that fatigue is related to both a central and peripheral factor. In summary, the brain can be subdivided into three parts: the brainstem, the cerebrum, and the cerebellum. The motor cortex controls motor activity with the aid of input from the subcortical areas. Prolonged endurance exercise can deplete excitatory neurotransmitters in higher brain centers and reduce motor neuron activation. This type of exercise-induced fatigue, i.e. central fatigue, results from the reduced activation of motor neurons and the muscle fibers they innervate. Now, looking at the motor functions of the spinal cord, we see the withdrawal reflex, as well as other reflexes important to the control of voluntary movement. There's also spinal tuning, which is the voluntary movement translated into appropriate muscle action. We see this in higher brain centers concerned with the general parameters of movement, and that details of movement are refined in the spinal cord. In summary, evidence exists that the spinal cord plays an important role in voluntary movement with groups of neurons controlling certain aspects of motor activity. The spinal mechanism by which a voluntary movement is translated into an appropriate muscle action is called the spinal tuning. Now, to discuss the control of motor functions, we see that subcortical and cortical motivation areas are involved. They send a rough draft of the movement. From there, we have the cerebellum and basal ganglia, which convert the rough draft into a movement plan. The cerebellum is involved in fast movements, where the basal ganglia is involved in slow, deliberate movements. Finally, the motor cortex is involved through the thalamus. This forwards the message sent down the spinal neurons for spinal tuning and onto muscles. Then we have feedback from muscle receptors and proprioceptors, allowing the fine-tuning of the motor program. Below what we see is a diagram with the structures and the processes leading to voluntary movement. In summary, control of voluntary movement is a complex and requires the cooperation of many areas in the brain as well as several subcortical areas. The first step in performing a voluntary movement occurs in the subcortical and cortical motivational areas, which send signals to the association cortex, which forms a rough draft of the planned movement. Next, we have a movement plan, which is then sent to both the cerebellum and basal ganglia. These structures cooperate to convert the rough draft into a precise temporal and spatial excitation program. From there, the cerebellum is important for making the fast movements, while the basal ganglia is more responsible for the slow and deliberate movements. What we see is from the cerebellum and basal ganglia, a precise program is sent through the thalamus to the motor cortex, which forwards the message down the spinal neurons for spinal tuning, and finally to the skeletal muscles. Following that, we have feedback to the central nervous system from muscle receptors and proprioceptors that allows the modification of motor programs if necessary. Now to discuss the autonomic nervous system, we see that it's responsible for maintaining the internal environment. There's effective organs not under voluntary control, such as smooth muscle, cardiac muscle, and glands. Within the autonomic nervous system, there's a sympathetic division which releases norepinephrine. It excites an effector organ, and after stimulation, the norepinephrine is removed from the synapse or inactivated. There is also a parasympathetic division which releases acetylcholine. This inhibits effector organs. After stimulation, the acetylcholine is degraded by the acetylcholine esterase. Here you will see a diagram of the neurotransmitters of the autonomic nervous system. As always, these diagrams for more detail are in your textbook. In summary, the autonomic nervous system is responsible for maintaining the constancy of the body's internal environment. Anatomically and functionally, the autonomic nervous system can be divided into two divisions, the sympathetic division and the parasympathetic division. In general, the sympathetic portion, releasing norepinephrine, tends to excite an organ, while the parasympathetic portion releases acetylcholine and tends to inhibit the same organ. Now, if we take a look at how exercise enhances brain health, we see that exercise improves brain function and reduces the risk of cognitive impairment associated with aging. Regular exercise can protect the brain against Alzheimer's disease as well as other diseases and certain types of brain injuries such as stroke. So how does exercise enhance brain health? What we see is an enhance in learning and memory, which stimulates the formation of new neurons, which would also improve brain vascular function and blood flow. It attenuates mechanisms driving depression and reduces peripheral factors for cognitive decline, such as inflammation, hypertension, and insulin resistance. Below you will see that exercise has a broad and positive benefits on brain health. In summary, research indicates that exercise can improve brain cognitive function, particularly in older individuals. With the content of this lecture being concluded, I want to provide you with some study questions to help you test your knowledge and to help you learn the information in more detail. Number 1. Identify the location and functions of the central nervous system. 2. Draw a simple chart illustrating the organization of the nervous system. 3. Define synapses. 4. Define membrane potential and action potential. 5. Discuss an IPSP and an EPSP. How do they differ? 6. What are proprioceptors? And give some examples. 7. Describe the location and the function of the vestibular apparatus. 8. What is meant by spinal tuning? 9. List the possible motor functions played by the brainstem, the motor cortex, and the cerebellum. 10. Describe the divisions and functions of the autonomic nervous system. 11. Define the term motor unit and innervation ratio. 12. Briefly describe the positive benefits of exercise on brain function. 13. How does regular exercise maintain neuronal health? And 14. Describe the withdrawal reflex. 3 more. We have 15. Outline the functions of both muscle spindles and the Golgi tendon organs. 16. Describe the general anatomical design of a muscle spindle and discuss its physiological functions. And 17. Discuss the function of the Golgi tendon organs in monitoring muscle tension. This concludes Chapter 7, the nervous system and the structure and control of movements. Please refer back to this lecture and your textbook to learn the information in more detail or to answer any questions. In addition, feel free to email me with any questions. Thank you.